is algae? Many of you probably think it's just that muck you have to scrape off the ponds, or that just covers the docks, your boats, that slimy stuff that people are just like, oh, I don't want it around. Algae is actually extremely important to the environment in natural conditions. It's the base of the aquatic food system. Many organisms eat it. They don't have roots. They draw their nutrients directly from the water column, or if they're benthic algae, from the substrate that they're living on. They can be attached or floating. Attached, we generally call them periphyton or benthic. Floating, phytoplankton. They can cause HABs, harmful algal blooms. A harmful algal bloom is not just toxic. It impacts anything from tourism, recreation, to aquatic organisms. So algae that smothers substrate and covers fish breeding habitat is classified as a harmful algal bloom. That covers our littoral zone, so makes it difficult for us to get into our swimming holes. That's a harmful algal bloom. Algae is used in wastewater treatments. If you go to any of the plants that have settling ponds, you'll see massive amounts of algae because it's there as a biological treatment. Many of you already know this and utilize it. Algae is used reliably to determine water quality impacts. Worldwide, let me specify that, worldwide, not just regionally, not just in the United States, worldwide. It is easily sampled and readily identified if you have a microscope. Sampling, you just collect from the area that you want, put it into the container, take it to the lab. It responds rapidly to nutrients, to the point where nutrients happen, then it is uptaken within days. They have known requirements and tolerance levels. It has been sampled, identified. Scientists from all over the world know how much nitrogen and how much phosphorus is needed for specific algae species. So if your phosphorus levels are still low and that algae is blooming like crazy, look to the nitrogen. Algae and biomonitoring. Biomonitoring, measuring biological responses to impacts using a living source. It's used worldwide. The European Water Framework Directive actually says use algae biomonitoring in littoral zones to determine impacts. It's written right into their standards. Many other countries across the world and states have documented practices for monitoring algae. Those metrics and protocols have been tested and confirmed. Both federal and state agencies, USGS, NRCS, EPA, Department of Agriculture, all have monitoring protocols and metrics. It can pinpoint pollutants. That is extremely important when you're looking at organic pollution, chlorides, heavy metals, or sediment. When you're looking at chlorophyll A, which is another surrogate that people use for sampling algae, that is just a water sample that looks at chlorophyll A, which algae use. All forms of algae use chlorophyll A. Some forms of algae will also use different forms of chlorophyll. However, chlorophyll A has been shown has a poor relationship to algae in littoral zones because with lower nutrients. And chlorophyll A only estimates biomass. Biomass, the abundance. If you look, there's a bunch of algae. Sample chlorophyll A, it shows there's a bunch of algae. You want to get down deeper into what the algae showcases. Algae can indicate the organic pollution from septic spills even when coliform testing does not. That means dilution, wave action, other things have pushed the coliform out and diluted it to the fact that you can't get that sample anymore. It has already impacted the algae because they've utilized those nutrients. Coliform also includes non-human sources. It indicates the excessive nutrients even when the water quality monitoring can't detect the phosphorus or nitrogen because algae utilizes it rapidly. Within days, it pulls it out of the water column and then proliferates. So when you're doing your water quality testing, you sample the water, that is a snapshot in time. Just right what is happening at that moment. The algae shows the history. It shows what's currently going on and what's been happening. Periphyton, what I really love, is the attached algae to the substrate, or the benthic algae. They rapidly uptake these nutrients before they can move offshore. So if you're doing offshore chemical monitoring, you're not going to have the chemicals there because the algae in the littoral zone is utilizing it. However, there does come a tipping point. That tipping point is reached when those nutrients far exceed what the algae can uptake. Then those nutrients move offshore to the phytoplankton populations, and that's where you can sample them, and it starts impacting the whole lake's biota. So the nearshore algae is a key indicator of what is going to be happening in a lake. So even after they diluted plants, it still shows it. And you can tell by just the dominant forms of algae that are present. You can tell by shifts in populations, sensitive forms of algae. And we're not just looking at diatoms. We're looking at soft forms of algae, the chlorophyta, the green algae, the yellow-green algae, the cyanobacteria, the blue-green algae, as well as the red algae. Organic pollution influences algae populations more than any other factor, more than temperature, more than light. It's the food source. That's what they need to grow. They will grow under the ice with no light if they have enough nutrients. Organic pollution is the biodegradable compounds that can be utilized by macrophytes, which are plants, and the algae. It will shift dominant forms of algae to those that are less nutritious for fish populations. Fish prefer more diatoms. They are healthier. Then it shifts the population over to the green algae, which is that filamentous long stuff that clogs intake pipes. They can wash up onto beaches and cause smell and odor and taste issues in your water. Then the next step is over to, it can do cyanobacteria, which eventually could cause a toxic algal bloom. And algal abundance can be traced to ammonia. Ammonia for algae is junk food. It's yummy junk food. And the pictures that are up on this slide right here, this is actually taken in a settling treatment pond. So yes, I did get in the pond to collect my sample. The next one is the sample in a settling dish. And then finally, it's not a cyanobacteria, it was actually a colonial green algae, and that's the picture of it, what it looks like under the microscope. So even if you see green water, doesn't mean toxic algal bloom. One of the key indicators that we use is called the Palmer Pollution Index. This is actually an EPA scientist who did a lot of research. He pulled over 165 authors, ranking species and genera of forms of algae that are specifically known for organic pollution in wastewater treatment. So from human waste. And these pictures up here, these are 3 forms that are identified on that list. Those are 3 types of algae. 2 of them are actually chlorophyta, and the middle one is a diatom. Using this metric, the Palmer Pollution Index metric, in combination with other metrics, you can start to pinpoint faulty septic systems, pinpoint sources of impact to water bodies. Some of the other metrics include the relative abundance and taxa richness, forms of cyanobacteria within the sample, as well as a few others that focus in on pollution, trophic, salinity, acidity, siltation, and then the similarity index. That one is very important because you need to have a reference site as well as a site that is completely impacted by that. That's why I get samples from the wastewater treatment facilities for those similarity index. This study has been happening in Lake George. We've been doing sampling within Lake George and the Adirondacks, focusing in on organic pollution, septic issues, and excessive nutrients, and chloride, which there is a relationship to chloride and phosphorus. Since 2008, the Palmer Pollution Index has been used to identify organic waste issues on Assembly Point, Oneida Bay, Silver Bay, Diamond Point, and Dunham's Bay. And the one in Dunham's Bay actually led to the creation of a wastewater management district, which will be discussed later. 1 year of data, 2014, over 79 samples within the littoral zone were taken. Of that, 15% indicated organic pollution present. And that was using the Palmer Index, indicating septic issues. 73 indicated trophic impaction from stormwater or something of that nature. 83% of the samples contained forms of cyanobacteria. Of those, 6% were actually dominant by cyanobacteria. In Dunham's Bay, we've been sampling there as well since 2008. That was conducted in the littoral zone to identify potentially faulty or under-functioning septic systems. Using the Palmer Index and other metrics, just 1 year sampling, it was determined that 5 of the sites fully analyzed indicated septic issues. We resampled in 2016. Since that time, the wastewater management district has been implemented and repairs and replacements have taken place. At those new sites where those repairs and replacements have taken place, we saw an improvement. There were lower organic pollution scores. So it showed that the science was there, that the implementation to fix is working. Within Lake George, we have been sampling after any spill to confirm our metrics. We were in the water after the Shepherd's Park spill, collecting samples. After the spill at the Sagamore, I was in the water collecting samples. And I will say the water quality sampling after Sagamore showed that it was all clean and clear. The algae said there is still an issue. We can say something happened here. It shifted the population within days. Samples have been collected regionally within wastewater treatment facilities and treatment ponds to further verify the metrics, to make sure that they are functioning. Not just within Lake George. I've been in septic systems and septic ponds within Vermont as well to make sure that this is not just a local metric, but that can be used regionally. Under the Lake George Septic Initiative, we sampled algae within the littoral zone this past summer in 2016. Of that, 2 samples indicated probable organic pollution present, and 2 indicated a lower level of organic pollution present. We ran regression analysis, indicated a mild correlation between the Palmer metric and total nitrogen. We are furthering this because we want more data sets to confirm. Under the pollution index, 4 indicated slight impact from pollution. Now that could be heavy metals, that could be detergents of any other thing that could get into septic systems. The trophic index, TI, 1 indicated severe impact, and 3 indicated moderate, and 7 slight impact. So that's trophic is more stormwater runoff, things that are going to be more upland surfaces up, people fertilizing their lawns. Then the eutrophic forms. 17 sites had 1 or more forms of eutrophic indicating species of algae present in the littoral zone, showing that we are having a shift in our littoral zone to a eutrophic area. Incorporating algal biomonitoring into any monitoring effort for lakes, streams, or ponds is a valuable tool. It actually can be cost effective because it eliminates the need to do other monitoring efforts. And if you have a trained scientist, you have the goal, you have everything right there. Using littoral algal sampling and these specific metrics is a proven tool to pinpoint sources that can determine water quality impacts, as well as to narrow down where you need to implement the fixes. It can also be used specifically for outreach and education. It is highly visible. People see algae, they know something is wrong, specifically when you have what's historically been a crystal clear lake. This metric and algal analysis is targeted for organic waste associated with septic systems and wastewater treatment facilities. It impacts change. It showcases what's happening in the water, even when water quality monitoring won't. And I would like to thank you and leave you with a brief picture of why I'm passionate about my work, because my son is passionate about algae as well. And this is him with his little microscope helping mommy run some samples. And thank you.